Welcome to video 6 of Genetics the Code Broken. In this video, we're going to be looking at the Human Genome Project, and we're going to be looking at some of the benefits, the limitations, and also how the Human Genome Project differs to linkage maps. So the Human Genome Project began in 1990 and aimed to identify all of the human genes, and there's about 30,000 of them, and to determine the sequence of the 3 billion bases in human DNA. It was expected to take 15 years, but in fact it was completed 2 years ahead of schedule in 2013. It was because that during this time there was rapid advances in technology in both molecular techniques and in electronics technology and computer software. So what were the aims of the project? So there was the aim of genetic mapping of the human genome, so identifying the location of genetic markers on DNA which are spaced across all the human chromosomes, to physically map the human genome, and that was done by cutting each chromosome into fragments and then determining the correct order of the pieces, to sequence DNA to determine the exact order of the nucleotides on each chromosome, and to analyze the genomes of other organisms and to provide a comparison and also to further understand their genomes to develop new molecular techniques that can be used for future research. So some of the benefits of the Human Genome Project was that it has advanced our knowledge and technology in both biology and medicine. And by having detailed DNA information, it's going to help our future scientific research. Within the medical field, it has improved the ability to diagnose, treat, and prevent some common diseases, and also to develop some new therapies for disease sufferers. It has provided a better ability to use family history to prevent diseases or to forewarn people of their genetic history. And by developing the technologies that were used during the project, they're able to use these for comparison between species, which will make further research possible, but also further research into gene expression will be made easier. And knowing the function of different genes will increase our knowledge of biochemistry and biochemical processes. So what were some of the limitations, since it was such a great discovery for scientists? Technology was originally seen as a limitation, but that was disproved as we saw finishing 2 years earlier than expected. All data is currently still seen as preliminary, and that's because now scientists are working at how can they further understand those so they can understand cell and organism functioning, and then how will they use that data in the future. It was relatively high cost, and some people question whether the results that we were able to achieve and they compare those to whether or not that money could have been spent to actually improving human health in the current state that it is. Because there were lots of genetic information being dealt with during this project, there's a lot of legal, ethical, and social issues that have risen from the study. That there's still some of the limitations is that the non-coding DNA differs in function between lots of people, and we still don't know about lots of the use of the junk DNA. And the Human Genome Project does not allow scientists to understand the role of every gene currently, or how it interacts with other genes, or how that DNA relates to behavior and other aspects of our brain functioning and neurobiology. So when we talk about the Human Genome Project, we're asked why we can't just use linkage maps to do a similar thing. And that's because genes that are mapped through linkage maps are coded for by an observable characteristic, but we know that lots of the human DNA is not coding, and therefore there's no observable feature or function to actually look at. And also, linkage maps only provide a relative position of genes on a single chromosome, whereas the Human Genome Project aimed to locate to an absolute figure or region the locations of the genes and provided the details of these order of the bases, which linkage maps currently do not. That's video 6. Make sure you tune in for video 7.